Welcome to Chapter 13, Notes Session Part 2. We've been exploring Mendel's laws or principles. What is this third law or principle? Well, previously we mentioned the law of dominance, where one allele is dominant over the other in expression. And the principle of segregation, where these alleles, these pairs of genes that we have get separated during meiosis. The principle of segregation, they're separating and going into different gametes. The third law or principle is known as the law or principle of independent assortment. So what does that tell us? Well, we know we have different genes for different traits. And independent assortment says they separate themselves independently of one another. And we will try to explore that visually looking at a dihybrid cross that Mendel used. So during meiosis, members of a pair of genes on homologous chromosomes get distributed into gametes independently of other gene pairs. So eye color gene separates itself differently than hair color gene. So again, how was this determined? How did Mendel figure this idea out? And it was his dihybrid crosses, testing for dominance relationships between alleles on two loci. Loci means location on a chromosome. So looking at two traits simultaneously, the probabilities for different phenotype combinations. Seems like a mouthful there, but when we see it visually, hopefully it'll make a little more sense. So what did he start with for his dihybrid cross? Obviously he didn't animate this slide. He started with two different pea plants. And one pea plant was dominant for the flower color, purple, and dominant pure for the height, tall gene variation. So the height gene, this is the tall allele. And the others were recessive for both trait variations. And so instead of purple flowers, they had the recessive white, little p, and little t, so they were all short plants. So F2, all the offspring then ended up being hybrid for both traits, because this parent can only give a capital P, this one can only give a little p, this one can only give a capital T, this one can only give a little t. And as a result, all the offspring will be dihybrid. And so that's the setup for a dihybrid cross. So then you're taking the pollen and pollinating the offspring that are all dihybrids to then make the next generation and see what occurs. Because the dihybrid plants all showed the dominant variations of the trait, even though they had the recessive allele hidden. So again, visually, you're crossing two different plants. Tall purple flowers, short white flowers. This is the pure recessive and the pure dominant. And then this is the dihybrid, this is the next generation. Hybrid for both traits. And then what are the gene combinations, or the genotype combinations for the two traits together? These are the possibilities in the gametes of these dihybrids. And so then you set up a four-sided Punnett square to try to reveal that. So one parent's genotype up here, the other over here. They're both dihybrids to start, so they have the same letter combinations here. Alright, so when we map it out, we're just, again, filling in the Punnett square as usual. And we end up with a 1/16th chance that you'll get a double recessive. But if you look at the filled in Punnett square completely, this is in your book, what is the chance that you're going to have white flowers and versus purple flowers? This is the same as if you did a monohybrid cross. It's 3-to-1 ratio. So it's still fitting the monohybrid, but when you're doing a dihybrid, what are the chances that you're going to get short flowers? 1 short, 2 short, 3 short, 4 shorts. 4 out of 16, so 25% time you have short flowers. So it's still sticking to the monohybrid cross. And if you were to do these Punnett squares separately for both traits, what are the chances that you get a short plant? 1 in 4. What's the chances you'll get a white flower? 1 in 4. So what are the chances that you get both together? 1/4 times 1/4 is 1/16th. So if you do them separately on separate plants through a monohybrid cross, the proportions stay the same. In other words, these different traits, flower color and plant height, are separating themselves independently of one another. So we ask ourselves, does independent assortment always hold true? And of course, in biology, there's always exceptions to rules, and this is no different. Mendel picked or noticed this pattern because he was looking at genes that do separate themselves independently, but some of the traits he looked at did not do that. And so the answer, of course, to this question is no, independent assortment does not always hold true. So it only works for genes that are on different chromosomes. If they're on the same chromosome, it's possible they might end up getting crossed over and separated together. So if you think of meiosis and crossing over, if they're close to each other on the same chromosome, they might end up getting crossed over together, and so then they would be considered linked. So it can occur between genes on the same chromosome if they're far enough away. So if they're far enough away on the same gene or same chromosome, there might be some independence in how they assort themselves, but no guarantee, depending on how much crossing over happened. So genes that have locations, loci, very close to one another on the same chromosome tend to stay together and not independently assort. And again, those are called linked genes. All the genes on one chromosome are called a linkage group because they have potential to be linked. The farther apart two genes are on a chromosome, the more often crossing over occurs, which could separate them more or less independently from one another. So the probability that a crossover will separate alleles of two genes is proportional to the distance between those genes. Thomas Hunt Morgan, when studying fruit flies, developed chromosome maps this way. So when we explore our virtual fruit fly lab, we will consider linked genes as an explanation of why we don't see the proportions or ratios that Mendel did. But I'll explain that a little bit later. So this is visually gene 1 and 2. They're close together on the same chromosome. They're linked. Gene 1 and 2 in the middle group, they're on separate chromosomes. They're not linked. And then gene 1 and 2 on the far right here, they're far enough away on the same chromosome that they probably won't cross over together. So they're probably not linked. Do Mendel's other principles always hold true for homologous genes? So dominance and segregation. So dominance is the one we're most interested in here. So again, Mendel focused on traits that were clearly dominant and recessive. But expression patterns for genes for some traits are not always straightforward. Can't answer all genetic questions with his 3 principles. Let's put it that way. So there's something called codominance. Codominance. Your book gives you some examples of codominance, but ultimately the 2 alleles both get fully expressed. And so if you have a heterozygote, neither is dominant or recessive in this case. You also can have multiple allele systems. A multiple allele system, genes with 3 or more alleles in a population. So our blood type is like that. Because we can have A and B, and they're codominant, but you also have type O blood. So there's a recessive in there. So it's a codominant situation for the A and B, but then there's also the recessive O, and so this is a multiple allele system. So your book explains what's required, what genotype do you need, and that kind of helps you see there's different phenotypes, more phenotype options when you have a codominant situation, or a multiple allele situation. Well, that would be true of the codominance as well. Alright, so if 2 alleles are not fully dominant over one another, that can happen too. So neither one is dominant fully over the other. They call that incomplete dominance. Incomplete dominance. One allele not fully dominant over its partner. And the heterozygote's phenotype is somewhere in between. It's kind of a blurring of the phenotypes, resulting in a 1:1 phenotype ratio of offspring. Snapdragons were the flower of choice for giving this as an example. So you can have red snapdragons or white snapdragons, but if you have them combine and have heterozygous snapdragon, it's going to be pink. And so again, with incomplete dominance, you'll see 3 phenotypes because you have 3 different genotypes. And again, one is not fully dominant over the other. So again, visually, that's what it looks like. Your book describes the crossing of monohybrids. It's going to give you this different phenotype look. It's going to not follow the 3:1 ratio that Mendel had with his truly fully dominant versus recessive traits. Our next session of notes is going to start off with this question. What is it called when more than one gene influences a trait? And that would be an example found if you discuss eye color.